Family Feud in South Africa, man, yeah. Oh, yeah, it's on and cracking. Let's do it then. We got the top 6 answers on the board. Points are double. Let's quit playing around. Here we go.
What is the function of the mitochondria?
E. Okay, Jesse.
ATP.
Wait, I thought mitochondria was the powerhouse of the cell. What could possibly go above ATP?
Actually, the mitochondria does much more than make ATP. It even plays a role in aging. Here's why. Let's go back to the beginning. Mitochondria are membrane-bound organelles that are found in almost all eukaryotic organisms. It is the cell's metabolic needs that determine how many mitochondria a cell will have. A muscle cell, for example, will have more mitochondria than a fat cell. Each mitochondria is surrounded by 2 specialized membranes that divide the organelle into a vast internal matrix and a thin intermembrane space. The porin protein creates many channels in the outer membrane, which function as filters to remove bigger molecules. The inner membrane is much more selective and consists of many infoldings known as cristae. The inner membrane makes use of transport proteins that will only carry molecules necessary to the matrix into it. The many compartments of a mitochondria can cooperate to produce ATP in a difficult multi-step operation. So to answer the question, what is ATP used for if not for energy production? ATP is all about information signaling. ATP hydrolysis, phosphorylation, and dephosphorylation all serve as signals to innumerable cellular pathways. What would it mean to have a mitochondria that didn't function? If a mitochondria can't maintain its proton gradient, loses the function of its electron transport chain, or can't obtain metabolites through its membrane, then its ATP production will slow or stop altogether. A cell needs ATP in order to transmit information and to function. A decrease in ATP production could cause the cell to function less efficiently and effectively, as ATP-dependent cellular pathways may be disrupted. If enough mitochondria are dysfunctional within a cell, the cell may cease to function entirely. Aging is the process of when a cell gradually declines in its functions and abilities. Another term commonly used is senescence. The difference between aging and senescence is that aging is a progressive decline, while senescence is a decline that occurs over the lifespan. Cells begin to stop responding to growth stimuli, stop dividing, and stop replicating. Essentially, the cell enters a permanent stage of cell cycle arrest. Why aging and senescence occurs, we don't really know, but there are key characteristics that contribute to it, including telomere erosion, DNA damage, epigenetic factors, and mitochondrial dysfunction. Senescence can lead to autophagy dysfunction, nutrient signal dysfunction, stem cell exhaustion, and chronic inflammation. Mitochondria play a heavy role in aging. Mitochondria can produce reactive oxygen species that can damage macromolecules within cells, which leads to the breakdown of DNA in the genome. This can result in senescence in the cell. The communication between the mitochondria and the nucleus break down early during aging. The mitochondrial membrane will deteriorate due to aging and releases DNA into the cell, which causes inflammation. This will also contribute to senescence. One thing many people don't know is that mitochondria are dynamic organelles that undergo constant remodeling by fission and fusion events in response to changes in stimuli. Fission is the division of mitochondria into 2 separate organelles. The main regulator of mitochondrial fission is the protein DRP1. Fusion is the process by which mitochondria fuse together to form elongated tubular networks. This is governed by proteins such as mitofusions 1 and 2 and OPA1. Mitochondrial dynamics play a key role in the regulation of cellular senescence. One key example is through pGAM5, which is a phosphatase necessary for fission through the dephosphorylation of DRP1. Studies have shown that pGAM5 knockout leads to increased mitochondrial fusion and decreased mitochondrial turnover, and exhibits markers of cellular senescence. This imbalance in mitochondrial dynamics leads to increased acute ATP and ROS levels and enhancements of specific signaling pathways such as mTOR. These signaling pathways are regulators of several cellular processes, notably mTOR being an inhibitor of autophagy. This ultimately leads to long-term decreases in ATP production and to cellular senescence. On the other hand, OPA1 decline has been observed in an age-related manner where muscle loss and disease is associated with OPA1 decline. Whether or not chronic OPA1 loss in muscle stem cells induces senescence is still a question scientists are working to answer. Therefore, as a whole, functional mitochondrial dynamics are critical to maintaining a healthy cellular state. Because of the role that they play in senescence, mitochondria could be the key to discovering anti-aging therapies. But that mystery hasn't been solved quite yet. Mitochondria really are more than just the powerhouse of the cell.
Well, thanks so much.